Before we jump into these phases of meiosis 1 and meiosis 2, and you're probably thinking, well, if we call them the same thing, how in the world do we know if we're in meiosis 1 or meiosis 2? Well, we're going to just put a Roman numeral after it. So we'll see that here in a second. But before we talk about that, let's talk about some stuff that can potentially happen. There are a couple different ways, because, I mean, we can have a combination of moms and dads genetic information. Okay? We can have the homologous pairs line up. But there's and that will give us, like, I can't remember the fancy pants words. I have it later, the number, I have it later on. And that gives us some degree of variation, but not a whole hell of a lot. What if I really want to spice things up here and introduce a ton of variation? Well, there are two things that you should probably be able to tell me about. And I don't know how many short answer questions I'm going to have, probably just a couple maybe on this exam. So you can already probably put it in your bank somewhere that I'm asking about mitosis, the phases of mitosis, explaining it. The other question can be from a number of different avenues. Okay, I can ask how meiosis, just how that variation can occur, and you'd be talking about this basically. Okay, I could talk about the central dogma of biology. That's probably the other really good one though too. Transcription and translation. That is in chapter 12. Okay. And that dovetails into chapter 14. So, I mean, I have chapter 12 and 14 are connected because of what we're going to talk about in chapter 12. And chapter 14 is just an application of that. Mitosis, chapter 9 is dovetailed in with this one. Okay. So explaining the differences between meiosis and mitosis, that would be a good question. Hell, I don't even have to ask you, put the phases of mitosis in the correct order and explain. I could just have you explain the differences between mitosis and meiosis. That may be a better question. Okay. For those thinking that there's no good question in the Mendelian patterns of inheritance, not so fast, my friend. Of course there is. So we have some good short answer questions coming up. All right. But I don't know if I'm going to ask all of them. So here, two ways that we can inject a lot of variation: crossing over and this thing called independent assortment. So let's look at these things. Crossing over, as you might imagine, it's just, there you go, crossing over. Pretty easy. I have a picture on the next slide that looks better than my two fingers. Okay. Crossing over involves the exchange of genetic material, DNA, between non-sisters. Okay. Between non-sisters. Non-sister chromatids. Okay. Remember, sisters. Let me color the other one. Different color, red. Okay. This is the bivalent. This is from mom and this is from dad. I'll put M and D. Okay. Mom and dad. These two are sisters. These two are sisters. These two are non-sisters. Okay. Non-cis. Okay. All right. So that's where we're right there. Non-sisters. Okay. I mean, there's a nucleoprotein lattice. I mean, yes. I mean, that's what helps to hold these two genetic strains together, right? Because this is just a gene, right? This is a chromosome. And even though we haven't gotten into it yet, shocking, if we start to kind of disassemble, okay, if I cut a piece off here and move it over here, this thing would have a tendency to unravel. Think of it like a sweater, okay? Once you get a string on the bottom of your sweater, if you try to pull it off, it just keeps coming and coming and coming. That could potentially happen to this chromosome. It could just unwind and then you'd be in a hell of a bad place, okay? But there are a number of proteins we're going to find, especially in chapter 13, where it helps to hold the structure of the DNA molecule together. And that's what this nucleoprotein lattice is talking about, okay? Am I going to ask a potential question about, what holds the synapses together? No, okay? Being able to describe crossing over, okay? When does it occur? Those are things that I'm probably going to ask about, all right? But what does this crossing over do? It exchanges genetic material. It gives me, excuse me, it gives me variation, okay? Because now what have I done? I've created a different sequence of adenine, guanine, cytosines, and thymines, right? Those nucleoproteins within the DNA structure. Okay, so here is my sisters, and these are my sisters. Okay, as you can see, they can come into contact with each other and touch. When they do that, there's a kind of lattice-like protein network that occurs around it, maintaining the integrity of the chromosome, and they just swap material. And there you can see it right there. Okay, this one and this one swapped, and I get a different sequence of amino acids now. And that could potentially give me a variation that goes from black hair to brown hair now, or to blonde. Okay. So it gives me a lot of variation. Gives me a lot of variation, especially if that's the sperm that, makes it to the egg. Okay. So we get an exchange of genetic material, and this gives us variation. And I don't know how many times it can synapse. I don't know if I have it listed on another slide. It can synapse tens of hundreds of times along the length of a chromosome. So it can do it a lot, and it can inject a high degree of variation. The last one is called independent assortment. Okay, independent assortment. This is just the chromosomes separate randomly. Oops, sorry. Okay. When the homologous chromosomes line up at the metaphase plate, it's not always this one goes here, this one goes here, this one goes here, this one goes here. Okay. They can separate in a random manner. They can line up at the metaphase plate in a random manner. All right. This gives me a kind of 2 to the 3rd or 8 different combinations between a maternal and a paternal chromosome. And this obviously, this mixing of chromosomes, as you can probably imagine, gives me a high degree of variation. What in the world does that look like? Well, we can have all mom on one side, all dad. Or we can have one flip-flop, or that position flip-flop, or that position flip-flop, or those two, or these two, or they can be flip-flopped on opposite sides. So there is a huge amount of randomness that occurs here. Crazy. We're also assuming it happens perfectly normal, okay? That they line up equally along that metaphase plate and that they'll separate just as equally. Obviously, that's not always the case, okay? But we're just talking about normal, everything is peachy fine. So I've got crossing over, and I've got this random assortment, independent assortment, where they just line up randomly. Mom, dad, egg, sperm come together and get you everything that you see in the mirror every single day. That is called fertilization. Okay. And there's that 70 trillion number there. Okay. 70 trillion, 368 billion, 644 million. Okay. If crossing over occurs once, because it can happen tens and tens and tens of times on a chromosome. If it happens only once, if you get one crossover occurring, one, you get 4 octillion, 951 septillion, 760 sextillion, 200 quintillion different genetically different zygotes possible. What the hell? That's crazy. That's crazy. This is what meiosis gives us. The big V, variation. Okay? Variation. This is so important when it comes to evolution. Okay? Giving us tiny little changes that accumulate and occur over the millennia that potentially gives an individual an advantage. Okay? And it can survive an environment better. Okay? Let's say something happens to that environment. Right? Maybe it's that one little genetic variant that existed that gave that individual an advantage to finding food, escaping, they were able to run just a little bit faster, jump a little bit higher. Okay. It is this right here that is going to give us all of the nonsense that's going to fuel evolutionary changes, is this thing called meiosis. That is 4 octillion, 951 septillion, 760 sextillion, 200 quintillion different zygotes possible. That is a absorbently high, crazy number, which will probably be our national debt here in a handful of years. So I guess we'll get more comfortable with it by then. Okay. But again, if it happens a couple, then I don't even know what that number is. I didn't type it into Google to find out how the hell to say it. Okay. I don't know. But this is what meiosis gives us. A huge amount of variation. Huge amount of variation. Because, there's a lot that goes into your brothers and sisters, right? If you all have brothers and sisters. You all look, similar, but there's just stuff that's different about you. Okay. On a multitude of various different levels. And it's just these little bitty differences. What's that? 4 octillion worth. 4 octillion worth of small little genetic changes that give you your uniqueness. You are all unique. Unless you're a twin, then you're not unique because there's somebody exactly like you in the world. Okay. So again, it gives us adaptation. It gives us variations. It gives us a potential advantage if the environment changes. All right. And that's the significance of this. Asexual reproduction, they're clones. Okay. Again, I need skin to become skin. I need liver to become liver. I don't necessarily need my offspring to be, exactly like me. Come on. I'd love to have my kids look and act exactly like me, to be perfect people, because I was perfect growing up. But, you want them to have an advantage, and hopefully I've given them something that that gives them that advantage. Some days I don't really know if I did or not. Okay. See, even Colton says it. There's just a little bit of difference. Okay. And we'll talk about this in the next, when we start talking about, evolutionary changes. All right. Asexual reproduction is fantastic if everything is the same. And by everything, I mean the environment. And the environment is never the same, right? There is changes occurring all the time. So when it comes to bacteria, they become more and more and more, but they can increase their number dramatically, right? They can divide every basically 15 minutes, some of them 20 minutes, some a little bit longer. Okay. But when it comes to, some of the stuff that causes us infections, some of the common infections, they can reproduce really fast. Okay. So they're able to increase the numbers, and it's that huge amount of numbers that allows them to overcome, obstacles in their environment, something that's changed in the environment that causes them to die off a little bit faster. Well, if they keep reproducing, there's going to be a variation that eventually occurs. And we see that with bacteria being, resistant to antibacterials now. Okay. But when it comes to giving us something that's an advantage, meiosis is the way. Okay. Meiosis is the way.
The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
So there you go. Now, if you're like my kids, then my kids, I have 2. Both of them look almost exactly like me. And that's bizarre, but they are not me, even though they have my youngest. Oh my God.